soon as a cell is born, the series of chemical events they are started, and these all chemical reactions that are interconnected, we consider them as cell cycle. For our convenience, we had discussed that the cell cycle is divided into interphase and mitosis phase, and then interphase is further divided into G1 phase and S phase and G2 phase. And we know that the central dogma run throughout the interphase, but the different phases of cell cycle, they have different properties. For example, in G1 phase, our genes, they give rise to RNA, and RNA give rise to proteins, but obviously all proteins are not formed in G1 phase, and mostly the function-specific protein are synthesized in the first part. Then the cell goes into S phase, which is synthetic phase or the DNA replication phase. In this time, the DNA is going to replicate itself so that during mitosis, the DNA is equally divided into two daughter cells. S phase is followed by the G2 phase. Again, now the DNA will be used to make RNAs, and RNAs will be used to make protein. But this time, the protein that will be synthesized are those that are involved in the process of mitosis. So once all the proteins are duplicated, DNA are duplicated, cell is ready for division, then it will enter into mitosis. And in mitosis, we have discussed in a bit of detail that first karyokinesis will take place, in which we will be dividing the chromosome quantitatively. Equal number of chromosomes will be divided, and this will be followed by cytokinesis, which is the unequal distribution of cytoplasm. And this unequal distribution of the cytoplasm will make this cell different from the other cells because of the concept of differential gene expression. So what we learned up till now is that when a chromosome is decondensed, genes will be exposed, and the next thing is, what is the use of these genes? These genes can be used in replication. These genes can be used for transcription, that is the synthesis of RNA. But if you have a very careful look on this beautiful diagram that is showing the cell cycle, you will learn that S phase is totally different than G1 and G2 phase. So what is the difference? In G1 and G2 phase, you are using the DNA, you are using the genes, but you are making RNA from them, and then ultimately you are making protein. While in the S phase, you are using the DNA that is exposed when the chromosomes are decondensed, but you are not using this DNA for RNA synthesis during S phase, you are using DNA for replication. What is the take-home message from this? The message is the DNA is used for replication, the DNA is also template for RNA formation, but both these things are not happening at the same time. This makes your old concept false, in which you always see a typical diagram of double-stranded DNA. On one strand, the picture shows that replication occurs, and on other strand, RNA synthesis occurs. So it means that that diagram is totally wrong, because both DNA synthesis and RNA synthesis never occur simultaneously. It means DNA will be acting as a template, but what DNA is going to do, whether DNA is used for replication or DNA is used for transcription, this is dependent on in which phase of cell cycle we are, and why the both processes are not occurring at the same time, why replication and transcription are not occurring at the same time, because we have learned that there are different enzymes for replication, there are different enzymes for RNA synthesis, and these enzymes are usually not active at the same time. So it means that the second level is very interesting, that when the chromosomes are decondensed, their nucleosomes are exposed the genes. Now the next question is, what will be the use of these genes? Whether the cell wants replication or cell wants transcription, this will be decided by when this chromosome is decondensed. In which phase of the cell cycle this chromosome is decondensed. If it is G1 or G2, the genes that are exposed will be used for transcription, and if the DNA is exposed during S phase of the cell cycle, now this DNA DNA will be used for the process of replication. We have already discussed in our last lecture when we talked about the structure of the DNA. We had discussed that the DNA is made up of specific structural and functional units that are called as DNA nucleotides, or more specifically, technically, they are called as 2-deoxyribonucleotides. In that discussion, we had discussed that a DNA nucleotide is made up of 3 units, a 5-carbon sugar, which is the backbone of the nucleotide, and then there is a nitrogen base, which is part of nucleotide, and we had discussed that there are 4 nitrogenous bases. They are adenine, guanine, cytosine, and thymine that are present in the structure of the DNA, but we know that one nucleotide contains 1 nitrogenous base. And the third component of the DNA nucleotide is the phosphate. And if you remember, we had discussed that the sugar molecule is 5-carbon structure, and all 5 carbons, they have some specific job to do. The carbon number 1 we know is the site of attachment for the nitrogenous base. Whatever is the base present in this nucleotide will be present on carbon number 1. The carbon number 2 is the differentiator, whether this is a nucleotide of DNA or RNA, because if you consider the name 2-deoxyribonucleotide, so this is clearly indicating that at carbon number 2, 1 oxygen is missing, and that is the major difference between DNA and RNA nucleotides. The sugar molecule in both cases is 5-carbon sugar, but in case of RNA it is ribonucleotide, and for DNA this is deoxyribose sugar, and the difference is 1 oxygen at the carbon number 2. So this position will differentiate whether it is a DNA nucleotide or RNA nucleotide, and this will be a key feature because the DNA polymerases will pick the DNA nucleotide and RNA polymerases will pick the RNA nucleotide, and they have slight structural differences. So carbon number 3 of the sugar and carbon number 5, we know they are the site of attachment. There will be phosphodiester linkage formation through which this nucleotide will be attached to the nucleotide above it, and also it will be attached to the nucleotide below it. So carbon number 5 is for attachment with the above nucleotide, and carbon number 3 is the attachment for the nucleotide that is present below this nucleotide, while carbon number 4 is the stable carbon having 4 valency, which is satisfied, so this is not going to do any specific job in the functioning of the DNA. So we know that DNA is a chain of deoxyribonucleotides that are attached with one another, and DNA is a double helical structure, so 2 chains of the DNA, they are connected with one another through nitrogenous bases. And if you remember, we had drawn the 2 strands and we had connected, and we had learned it already that the 2 strands of the DNA are anti-parallel. We also have a concept of 5' end and 3' end, because we know that the first nucleotide in a chain of nucleotide has no nucleotide above it, so the 5th bonding position will be written, and at the end of the DNA chain, the last nucleotide do not have any nucleotide after it, so its 3rd position will be written for the binding. So that is why whenever we see the structure of a DNA, we always see 5' end on one side and 3' end on the other side. As the 2 strands of the DNA are anti-parallel, so 5' end of one strand is facing the 3' end of the other strand, and vice versa. So this is what we already know, just reminding you so that we can understand the process of DNA replication. So coming back to today's topic, so today we are going to discuss that when a part of chromosome will decondense, now the genes will be exposed. So what will happen at this level? So now we are starting our 2nd level of regulation, that is what happens at the level of DNA. So if you remember, I had discussed in the beginning when we talked about carrier stereotype that we have a 46 chromosome and we have about 25,000 genes, and we learned that one chromosome contains hundreds and thousands of genes, and the number of genes varies from chromosome to chromosome. Now, just to give you an idea, in as usual very simple and basic manner, imagine that a part of chromosome is decondensed. So now DNA is present in the form of genes that will be exposed. Now, first thing is that the genes that are exposed, they are present in a stable form, right? Because the two strands of the DNA, they are connected with one another. So point one is that there are many genes that are present in this region, which is decondensed. So theoretically, these genes are available for the next level of regulation. And as we are talking about S phase, because we want to discuss DNA regulation at the level of DNA. So theoretically, there are many genes that are exposed and they can be used for replication, but technically none of these genes can be used for replication. Why? The answer is very simple, because these genes are present in a stable form. The two strands of the DNA, they are attached with one another through hydrogen bonds, and until these hydrogen bonds are existing, the two strands will remain connected, the valency remains stable, and until the DNA is stable, obviously we cannot use them for any process including replication or transcription. So it means that only decondensing the chromosome will not solve the job, will not do the trick. But when you decondense the chromosome at level 1, you are exposed to many genes, and the genes are present in a stable form. So first thing that we learn, that we need to unstable this DNA. I will be discussing it in a minute. But I want to introduce another concept which is equally important. So one thing is that this DNA is stable, the genes are stable. You have to destable them, you have to separate the two strands of the DNA so that their bonds are broken, the valencies will be unsatisfied, the ionic situation will create, and now the DNA will be reactive, and only then the enzyme can do replication in this case, or transcription in case of G1 phase or G2 phase. But another important thing is that the region which is now decondensed, or the chromosomal part which is decondensed, that contains several genes on them. And one of the key features that we learned in our physical classes was that genes are diverse. Genes are all made up of DNA. They are all made up of deoxyribonucleotide. But how genes are different from one another? They are different from one another because they have different number and sequence of the nucleotides. So obviously a gene which is consist of less number of nucleotides is different than a gene which is consisting of more number of nucleotides. But there is another angle of the story as well. One thing is number of nucleotides, that is varying from one gene to the other. The second thing is sequence of the nucleotides. Remember, I had given you an idea of a base pair, that the two strands of DNA, they are attached with one another through hydrogen bonds between the nitrogen spaces. And we had learned a rule at GC, at Government College, that on one strand if there is adenine nucleotide, it will bind to the thymine nucleotide on the other strand. And we had learned that this AT is one base pair, and if there is a GC, this is another base pair. So it means DNA is present in the form of base pair. Why I have reminded this thing again to you? Because whenever genes are exposed on a decondensed chromosome, they are different not only in the number of the nucleotides, they are also different in the sequence of nucleotides. Remember, we also have a concept of open reading frame, that every gene has a specific sequence of the nucleotide that are converted into RNA nucleotides, and then it is converted into specific type of protein. It means every gene has different number and different sequence of the nucleotides. So it means all the genes that are exposed on this decondensed chromosome, they are not same. So if all the genes that are exposed, they are not same regarding number and sequence of DNA nucleotides, then how they can be handled at the same time? So the level 2 says that you are exposed with lot of genes and you are in S phase, so you want to undergo replication, but you cannot undergo replication of all genes at the same time. Why? The answer is because the composition of all these genes is not same. And how the genes they differ from one another? The genes differ from one another in the form of base pairing. And what type of base pair they have? So as I mentioned in the last clip, that we have two types of combinations in the structure of DNA, that adenine and thymine, or thymine and adenine, they bind with one another and there are two hydrogen bonds present between them. And also the second option is guanosine binds with cytosine, or cytosine binds with the guanosine, and they bind through three hydrogen bonds. So one of the criteria to study the gene structure is the AT contents and GC contents of a gene. So AT content is showing is what does it mean? How many A and T combinations are present in a gene structure? And GC content means how many G and C bonds, how many GC base pairs are present in a gene structure. So I give you two simple examples just to give you an idea that the genes that are exposed on a specific part of decondensed chromosome, they are not same. So look at this simple handwritten diagram. You will see example A and example B. Both are showing stable DNA because the two strands are connected. Both are showing 3' end and 5' end, and antiparallel strand is showing 5' end and 3' end. In a first bird eye view, you will simply say that there are DNA nucleotides that are connected with one another. But if you have a concentrated look on the example A, you will be seeing that in this there are total 9 base pairs, AT, GC, AT, CG, TA, CG, GC, AT and AT. So there are 9 base pairs, and you can see there are AT and GC, both combinations are occurring. And in the B example, you again can see there are 9 base pairs that are existing, but if you have a careful look on the two genes, you will see that the number of nucleotides is same in both cases, but the sequence of nucleotides is not same. In the example A, you have 5 out of 9 combinations that are A and E, and you have obviously 4 out of 9 combinations that are G and C. So for this gene, you have more AT contents, you have more AT base pair than GC base pair, and such genes are considered as AT rich genes because they have more AT than GC bonds. And in example B, if you have a look, the number of base pair is 9, but the AT combination is just 2 out of 7, while GC combinations are 5 out of 7. It means that both genes have same number of base pairs, but the composition is not same. Example A gene is AT rich gene, but example B is a GC rich gene, simply because they have different nucleotides. But technically, how they are different? What is the other difference? If you have a look on the A example, you have more AT region, and A and T has 2 hydrogen bonds, while example B is a GC rich gene which have more 3 hydrogen bonds, and just very simple calculations, it is easy to break 2 hydrogen bonds, and it will need more energy to break 3 hydrogen bonds. So very simply, with this simple example, I just want to give you an idea that when a chromosome is decondensed, many genes are exposed, but all these genes have different combinations. The first thing is you have to unstable these genes, but obviously you will need different energy to unstable the gene which has more AT bonds, and you will need a higher energy to unstable the gene which is more 3 bonds that are GC rich region. So with this simple example, we are concluding that all genes that are present on a decondensed chromosome cannot be handled at the same time because they have different composition.